Assalamualaikum. In this video, we are going to present about two activities that we have been doing in our BIO330 lab. The first activity is the Capture-Recapture, and the second activity is the Quadrat sampling technique.
Hi everyone. First thing first, let's see what is Capture-Recapture. Capture-Recapture is a sampling method that we use to estimate population size. To undergo this method, we need to set up a controlled investigation where the object, commonly animals, are captured, marked, released, and recapture it again after a period of time. The proportion of the marked members in the size sample can give an estimate to the population size.
To begin a capture-recapture experiment, the first step is to determine the size of the study area, ensuring it is well-defined and appropriate for the species being studied to enable effective sampling. Next, we use traps to capture a group of individuals alive and mark each with a unique identifier such as non-toxic paint, numbered tags, or bands. Then, we release them back into the environment unharmed. After making and releasing the individuals, we must allow sufficient time for them to redistribute themselves among the unmarked population, which depends on the species mobility and behavior, ensuring they mix thoroughly. Finally, we return to the study area and capture the individuals alive once again, recording both the marked and unmarked individuals to estimate the population size by comparing the number of marked individuals. Here is the video of 5 of us doing the capture-recapture sampling technique.
Now, let's see the estimate population. The equation is n equal to m multiplied by c over r, where n, estimate of total population size, m, total number of animals captured and marked on the first visit, c, total number of animals captured on the second visit, r, number of animals captured on the first visit that were recaptured on the second visit. For example, some of the individuals in the second sample would have been marked during the initial visit and are now known as recaptures. Other animals captured during the second visit will not have been captured during the first visit to the study area. These unmarked animals usually are given a tag or band. Population size can be estimated from as few as 2 visits to the study area. Commonly, more than 2 visits are made, particularly if estimates of survival or movement are desired. Regardless of the total number of visits, the researcher simply records the date of each capture of each individual. The capture histories generated are analyzed mathematically to estimate population size, survival, or movement. Next is how to calculate. The Lincoln-Patterson method can be used to estimate population size if only 2 visits are made to the study area. This method assumes that the study population is close. In other words, the 2 visits to the study area are close enough in time so that no individuals die, are born, move into the study area, which is immigrate, or move out of the study area, emigrate, between visits. The model also assumes that no marks fall of animals between visits to the field site by the researcher, and that the researcher correctly records all marks. Next, this is a refined form, which is a slightly better estimate of population size can be obtained with a modified version of the first formula. This modified formula reduces bias in the population estimate as shown as below, whereas before, n, estimate of total population size, m, total number of animals captured and marked on the first visit, c, total number of animals captured on the second visit, r, number of animals captured on the first visit, that were then recaptured on the second visit, Therefore, an approximately unbiased variance of n over n can be estimated as the equation below.
So, based on grasshopper sample, the total of first and second capture is 18 and 14 grasshoppers. The grasshoppers that has a mark on their wings is 7 grasshoppers. Next, on the second samples, which is butterflies, there is 2 groups. The total of first and second capture is 13 and 18. And the butterflies that has a mark on their wings is 14 butterflies. So, by using the Lincoln-Peterson method, we have found the estimate of total population size for both grasshoppers and butterflies. In this experiment, the Lincoln-Peterson method estimates that there are 36 grasshoppers and 59 butterflies in the grassy area around UiTM Jenkins Library.
So, in conclusion, the Capture-Recapture method is a valuable technique for estimating population sizes in ecological studies. Our investigation estimated the population of species in study area at approximately estimated population size, consistent with prior knowledge, while assumption like a close population and equal catchability may introduce errors. Factors such as migration and behavior responses also affect the accuracy. Future studies could use advanced models or larger sample sizes to improve reliability. Overall, this method provides crucial insight into population dynamics and supports informed conservation strategies.
And on to the next activity, which is quadrat sampling technique. I'll be explaining about what is quadrat sampling technique. First of all, what is quadrat sampling? Quadrat sampling is a method by which organisms in a certain place of the habitat are counted directly. This quadrat method has been widely used in plant studies. And this quadrat method has been used to estimate population abundance, density, frequency, and distribution. So, I'm gonna briefly explain to you guys on how to use the quadrat sampling technique. So based on the example on your left, the quadrat positions are chosen randomly so there are no bias detected. Or they can be placed along a transect. Transect is a line placed across a community of organisms.
Let's look at quadrat sampling. There are 2 types of quadrat sampling, which is systematic distribution of quadrat and random distribution of quadrat. Systematic distribution sampling technique is a statistical sampling technique that we use to select a sample from a larger population in a systematic way. Systematic sampling involves choosing every KTH element from a list of population, where K represents the sampling interval. Random distribution sampling technique is a technique to select a subset of individuals from a larger population, where each individual has an equal chance of being chosen. This method ensures representative samples, minimizes bias, and allows for reliable inferences about the population based on the sample data.
Next is procedure for quadrant sampling technique. The first one is systematic sampling procedure. Quadrants are placed at the same intervals along transect which run across the investigated area at the same intervals. Second is random sampling procedure, using random number table. Next is the procedure for a sampling technique using line transect. The first one, determine a baseline along the border of the area under investigation. Second, choose a series of points along this baseline either randomly or systematically. Next, these points are used as the starting points for the transect to run across the area being investigated. Then, record only the plants which touch the line as seen vertically above or below the transect line. Therefore, 10 to 20 lines are placed randomly in the area to provide enough samples to investigate the community.
So, as you can see, this is the result of our findings using quadrat sampling methods. The species include Decantium, Gruna heterophylla, Tridex procumbens, and also Pectis prostrata.
In conclusion, quadrat sampling is a valuable method for studying organisms within a specific habitat by counting them directly within defined areas, known as quadrats. This technique is particularly effective in plant studies and has been extensively used in many projects. By placing quadrats randomly or along a transect, researchers can assess the abundance and diversity of organisms. Systematic distribution sampling, a complementary technique, involves selecting samples at regular intervals and offers efficiency and ease of implementation. On the other hand, random distribution sampling ensures each individual in a population has an equal chance of selection, reducing bias and enhancing the representativeness of the sample. Both systematic and random sampling methods have their own advantages and disadvantages, but when applied correctly, they provide reliable and valuable data for ecological studies.